Hello, welcome to the Harry Garegin Show, where we dive into the tech-driven future of healthcare. Investors and companies in the life science industry have been betting a lot of money over the last few years on a single idea. If you boil it down to the simplest terms, it's the idea that computation will help us get a lot better at developing new drugs. The argument is that as we get more and more data about the DNA and RNA sequences of single cells and whole organisms, we can use software to figure out exactly what's going wrong in various diseases, and then we can apply computational design or discover new drug molecules that will interrupt those disease processes just at the right moment and prevent or repair problems inside cells without any bad side effects. But the word computation covers a pretty broad range of techniques. And the reason that there are dozens, if not hundreds, of computational drug discovery startups popping up is that everyone has their own hypothesis about what specific kind of computation is going to be the most powerful. For example, you might be convinced that the most important thing is to understand the physics of protein-protein interactions at an atomic level. And so, you would put your money into atomic scale simulations that show how proteins fold or unfold to form different shapes under different conditions. Or, you might think that it's more important to model proteins at the molecular scale to make predictions about whether and how a particular drug molecule might dock with a target protein. Or you might think that it's smarter to try to model whole cells and see how different molecular pathways interact to affect different functions of the cell. Or you might not care about the details of physics or chemistry-based models at all. In that case, you could just take a big generative AI model, similar to a large language model, and train it on a huge amount of unlabeled data about genes and proteins in diseased cells and healthy cells to see what kind of predictions it comes up with. It's too early to say which of these computational approaches and which level or scale of focus is going to be the most fruitful. But maybe you don't have to choose. Maybe you can bet on all of these different ideas all at once. My guests this week are the CEO and CSO of a startup that's taking an all-of-the-above approach. It's called Deep Origin, and it was formed last year from the merger of two companies founded by theoretical chemist Garegin Papaoian and software builder Michael Antonov. Antonov helped found the virtual reality hardware company Oculus. After Facebook acquired Oculus, he got curious about longevity and how software could help untangle the trillions of gene-protein interactions that mediate health and disease. He founded a company called Formic Labs to dig into that problem, and last year the company changed its name to Deep Origin. Papaoian is an academic scientist who also took the helm as CEO of his startup Biosim AI, who's interested in how to use software to model molecular dynamics and quantum chemistry. Recently, Antonov and Papaoian decided to join forces, and Biosim AI merged into Deep Origin. And they say the company's philosophy is that physics-based modeling by itself won't be enough to build a powerful drug discovery engine. But neither will generative AI, which requires more training data than lab scientists will ever be able to provide. So they think the only reasonable approach today is to combine the two and use both physics and AI to try to get better at predicting which molecules could become effective drugs. Exactly how Antonov and Papaoian came to their conclusion and how the integration is playing out was the main theme of our conversation. It's important stuff, because if Deep Origin is right, then a lot of other, more specialized biotech and tech bio startups could be going down the wrong path. So let's dive in. Michael Garegin, welcome to the show. It's great to have you both here, and I feel like I've been thinking about this, having you guys on the show for a long time, but I'm glad we finally got it scheduled and got you guys on.
It's a pleasure, Harry. Thank you for inviting me.
It's a pleasure. Happy to be here.
So I want to, I guess, give everybody sort of at least a level set on your backgrounds, because Michael, like you did not start in drug discovery and biological sciences, if my homework is correct. So can you start like a little bit about your background so people can understand and how you ended up in this space?
Yes. So I've actually originally was a software programmer, very passionate about computer games. So built two, was a CTO, technical lead in one of the game technology companies, and ultimately was a part of the Oculus VR journey, which where I did lots of infrastructure SDKs for the Oculus Rift, computer vision, other technology stack. And so for many years, I was really into building software platforms. And ultimately, when I was in, actually, we joined Facebook after the acquisition, I was trying to imagine what is the most meaningful thing I could do with my life. And I got very interested in longevity. And because the genomic revolution has happened, there was a lot of noise. And I was curious about just how much do we understand it. And the other part, which became very interesting to me, is how do our bodies work? So I ended up taking lots of courses. I remember biochemistry was like the hardest thing ever while working full time, sitting with pre-med students. And that's probably the type of thing which is also drove Garek's passion, is being able to see at that point animations like of the ATP and how all this cellular machinery works like in small microscopic world. It seems predictable, it seems something we can understand, and that got me very interested in simulations, but also what can we do for longevity. So long story short, I've started investing in a space very cautiously. I realized it's quite risky, and learned, and ultimately decided that we need a lot better scientific tools. So you imagine even like a single cell can have 10,000 expressed genes. There's trillions of them, all the interactions. There's no way a human is going to be able to get it and understand it. And if we're going to truly improve health span, we need to be very kind of multimodal, attacking many pathways and things at once. And the only way you're going to do that is you can simulate or understand the system. So I've really dedicated and decided that a lot better software and infrastructure needs to be built to speed up this process.
Well, the one key takeaway I'm taking away from this is it's never too late to go back to school and start a whole new career in a whole new space. So for anybody that's listening, think about that. Garegin, I mean, you're more, I want to say grounded in the space, but I know like you're super interdisciplinary. So like a unique background of components that are coming together to attack what you're trying to attack now. So can you go a little bit into your background a little bit?
Yeah, absolutely. So I'm a theoretical chemist, and I've been most of my career, and I'm still in academia part-time, or on a part-time sabbatical. Theoretical chemistry is itself an interdisciplinary area that sort of is part chemistry, part physics, and in the last 40 years, it's also become part computational science, where we have to also program. For those of us that are quantum chemists, that you program quantum chemistry codes. If you do molecular dynamics, then you program molecular dynamics codes. And just to give sort of a perspective on how complex these things have become, one of the leading molecular dynamics codes in the world is called GROMACS, and it has 2 million lines of C++ code. So it's a humongous project with lots of moving parts. Probably no one fully understands how it works at this point. It's just so big. And so it sort of requires not only just knowing equations of chemistry, equations of physics, but also what are the best software practices? How do you develop a complex software product and maintain it, and documentation? So I've been, and you're right, so I've been interested in not just in chemistry, but in physics, in computer science, and over many years, I've done some paper and pencil papers that I enjoy, just analytical papers. But most of my work has been computational, in computational chemistry, computational biology. And I feel that I enjoy, and my strengths are being a tool maker in this space. So I like building complex new tools in computational chemistry and biology, and then applying it to novel scientific problems. And a few years ago, I realized that over my career, I participated in many projects, but they have this dissipation tendency that after you move on, then this software decays and maybe it's not maintained well and so on. So there was an idea that I bring my experience in developing new tools in computational chemistry to work on problems of drug discovery. So that's how I launched Biosim, and then pretty early on met Michael, and we started early on working closely together, having shared vision, and also helping with each other in many ways. And yeah, so that's roughly how it started. So and I feel that the startup a startup company allows many new opportunities for this software development, and that can applied both to health and longevity that in academia would have been hard to accomplish. So yeah, so it's a new life for me too, actually, in a way.
Yeah. So, I mean, for everybody that's listening, I mean, I've been watching these companies for almost since inception. So, been watching how they've evolved and grown, but I think you guys have had to put a unique team together, not just say what I consider drug discovery and say AI, but a real understanding of, as you said, Garik, was artificial intelligence, molecular dynamics, physics, and a number of other disciplines to sort of bring something together that then can get you to the answers that you're looking for. I mean, how do you look at these two fields intersecting in your approach to drug discovery? What is the advantage of sort of this interdisciplinary expertise offer to that, say, a normal approach may not?
Mm-hmm. Yeah, definitely. Great question. So historically, until this recent revolution in AI, the approach in computational modeling and simulation was physics-based. And similar to, for your listeners, to make an analogy, similar to maybe weather modeling, that, like every day you open your iPhone and it sort of says, oh, tomorrow it will rain, or maybe even in a week. And the question is, how can they predict that, right? And it turns out there are these super complex simulations run on huge supercomputers that take the whole Earth and atmosphere and put it into small grids and just sort of model how air moves from place to place and heat transfers. And from all that, they can then locally for you tell you that you'll have snow tomorrow. So it's pretty remarkable that they can do that. But there are limits to, for example, you cannot predict weather for the next year. Like, there are limits to how far into the future you can predict. Like, even though you have the physics equations, but they sort of degrade, in a sense, the predictions degrade if you go into longer time horizons. So there are limits to physics-based modeling. There are similar limits to physics-based modeling in biology and chemistry, is that there are some things that we can do a really impressive job and get nailed answers, and you check it with experiment and you get to the 4th or 6th digit accuracy in terms of the result you predicted and the results that experiment measured. But then typically, especially in biology, most things we cannot predict that well. Proteins are complex, DNA is complex, and then so they all come together and form this sort of mess that's very dynamic. And it turns out that purely physics-based modeling has really hard time to just go and photorealistically model these things. Maybe 100 years from now it could, or quantum computers, I don't know, like at least for the next few decades, that's out of the question. And then the AI came along, the AI revolution 10 years ago, and I'd say I feel that there was maybe a bit of a, so there was this huge shift that, especially in the industry, but also in academia, but largely in the industry, where lots of physics-based modeling was forgotten quickly, and everyone just wanted to take data and just put AI on it and quickly get low-hanging fruit. And it was appealing to investors. It was appealing to, like, we can just use the magical AI and get something done in a month that was unsolvable. So that's the, in a sense, that's the situation today to a certain degree. But I feel that, again, in biology, you'd appreciate it, that data is like every data point you get through sweat and tears and money. So it's not easy to get like 10 million data points for your AI to have plenty of data. In some problems, you need more than 10 million. Maybe you need 10 billion data points, and it's completely out of question. So that's where I think we are forced to sort of reckon that you cannot just do, in some problems you can do, maybe there is enough data, maybe the problem is narrow enough. But in most problems in biology and biomedical research, you somehow have to combine the two. And that's my philosophy, and I believe that's Michael's philosophy.
Yes,
Yeah,
that's
that's interesting.
actually,
Go ahead, Michael. Sorry.
it is part of the reason we have these kind of two components to the company, is because we realize whatever is done in simulation, it needs to have experimental validation as a part of it, but also certain things you do need to have lots of software infrastructure to even track experiments. I think the state of the art in many areas today is that you do have databases and you may have limbs and certain things to know a bit about your lab, but at a certain level, your lab, your software does not actually know and understand what is the data it captured. So somebody may put in a spreadsheet with like a result of plate readout for different, like what are the different things in each well for each cell type, but they don't know what the columns mean. Therefore, software cannot come in easily and interpret it. So lots of bioinformatic work needs to be done. So that aspect is not going to go away. Hopefully, it'll become more optimized with robotics, simplified. So at the same time, these two systems need to be married. So if we are going to fully close the loop on this integrated, wet simulations to wet lab to understanding cycle, then we need software which looks at all of these parts and makes it accessible. And that's really the way we look at it. I'm really excited about the simulations and really kind of AI and physics bridging the gaps for broader models. I hope, like, one thing I'd like to talk to Garrick about is this molecular self-assembly, which is that I define it a slightly bigger problem. So because you have a protein folding, you have interactions, which maybe MD handles, but can we actually throw a volume of molecules with DNA, RNA, and everything in a soup and have it do what this biology does? Can that be done within, say, the next 5 years? And clearly that won't be just physics. It needs to be physics and AI and things. But if we can really do that, then we can truly understand biology. So kind of marrying that and supported by experiments is very exciting.
Oh yeah, I mean, I love this space because there's not a day that goes by where I'm like, either I don't know something and I got to learn, or I'm learning something new, which is what keeps the space exciting. But I've talked to lots of people on the show. I've talked to lots of people, new companies and so forth. What makes the approach that you guys are taking to drug discovery, say, unique compared to, and I'll just say traditional methods, not to pull all the new stuff into play? What's the twist? What's the secret sauce?
Yeah, I think because we have both the simulation part and the life sciences platform, we probably want to have two answers to that question, and then the totality of them coming together. On the simulation side, I think our philosophy is that we need to develop new tech and that can advance the field, and in particular integration. That, you know, if you think about a cell, you say, okay, a cell has billion proteins, but then you zoom in, and then you have maybe 2-3 proteins coming together in a complex configuration, and then you zoom in and there's a protein pocket, and you start to see side chains and they are rotating, and you start to see hydrogen bonds. Well, it turns out that you at least in today's world, you cannot have a single model that describes all that. Although it's a single reality, but just for like, as again, the same thing as we do, you do local weather modeling, you do maybe for the country, you do for the planet. Like these are different models that maybe talk to each other. It's similar for cells too, is that we have a model that maybe with quantum chemistry models fine details of how hydrogen bond is made between two small molecules. And then you go to the protein level, that's another set of models. And then you go to the cell scale, it's completely another set of models. And so most companies typically focus just in one of these spaces. And because of that, they sort of miss, like you might focus on the pocket, but sort of not see, oh, there's a big protein coming. And because of that, your pocket changed. And the calculations you did sort of doesn't make as much sense because you ignored that interaction and so on. So one thing that I think distinguishes us on the simulation side is that we have 4 teams working on 4 different scales, from small molecule docking to molecular dynamics of proteins at atomistic scale, then coarse grain modeling of multi-protein assemblies, and then whole cell modeling. And then and these teams then can work with each other, in projects to make sure that there is consistency, and then bringing AI to all that. So I think that technology stack, the depth of it or the breadth of it, is something that I think quite unique. And the combination of physics and AI is powerful too. So we think that we are developing a powerful new overall technology stack.
Yeah, I was looking at some of the slides that you sent me, and thank you, because that completely changed every question that I had that I originally came up with. So it brought me up to speed on where the company is. But you had a couple of slides there where you're saying the AI enables much more faster and say accurate virtual screening. And so is that a function of the code? Is that a function of the tech stack you guys are using? Is that a function of how you've assembled the pieces to get you there compared to some of the other systems that you had on there? Just curious of that technical advantage, because I assume it means less GPU time and therefore a lower cost.
Yeah, again, in that case too, Harry, it's a combination. It's not pure AI. We cannot actually get these results with pure AI, meaning if we just come and say, we don't know physics or chemistry and we don't want to know, we'll just throw the data into like some super powerful AI and it will all figure it out for us. I don't think we could get results like that with that approach. So it is heavy physics there too. But then we sort of know in important places where physics has gaps, we sort of plug those gaps with AI. And then it becomes almost like an artisan work. You just have to know that what are the strengths of physics, but also what are the weaknesses, and where AI can glue in, and they can, because sometimes they might not be completely compatible with each other. So it's a very artisan work of basically assembling the architecture, let's say, of that virtual screening algorithms and software. And a part of that is the question of, we have really strong teams in these areas, very talented folks, and I think that's part of they learn and have background in sort of having that artisanship in building software like that. Yeah, so I think that would be my answer. That just how there are infinitely many ways how you could combine AI and physics. Even in AI, as you know, there are many ways. Like there are many AI approaches. Some of them are better than others, and then there's also lots of data. There are also lots of data issues, like what data do you take? How do you clean it? Like lots of sort of small questions that maybe each of them is not a huge thing, but if you string it together, then you even sort of two similar teams working on a similar problem could end up with very different solutions just because they answered those questions differently along the way.
So, you know, I'm going back to the first conversation we ever had, and I don't think the premise has changed, but this whole concept of undruggable targets, and how is it that the system that you two have created or brought together enables better drug design to tackle this challenge? And just to add on top of that, right, I think, how do you identify high quality hits when you're doing that? So say, you know, maybe you could both tackle the answer to that combo question.
I could maybe go first. So I'll start with the second one. Obviously, the ultimate way to check whether you have a hit is by experiment. So you could have an in vitro or an in vivo experiment and have an assay and test and see if your computational predictions come true. You could synthesize a molecule and then bring it to, let's say, a cell assay or a binding assay and see if you see the outcome that you're expecting. Interestingly enough, to even there, computation can help. At the end, you still need an experiment for sure. That's the ultimate answer. But as sort of as an interim step, there are these computations called free energy perturbation in molecular dynamics that, for example, in drug binding, you have your protein, you have your putative drug in the pocket, and then you have water around it and ions and metabolites, anything you want. And then you could do this advanced calculation in molecular dynamics where you say, what's the binding energy of that? And it turns out that that if it's not an easy calculation to do, and computationally it's expensive too, but you might be able to get an answer to 1 to 1.5 kilocalorie accuracy. That, for example, for heat discovery stage, when you are looking at many virtual molecules, that could be good enough, and it might allow you to, when you select computationally molecules that way, when you go to the experiment, then you get really high hit rate in terms of the experiment. So that's maybe I'll stop there. We can come back to the undruggable targets. Let Michael chime in.
Well, actually, I know it's a little bit of a side comment, but one thing which got me really excited working with Garrick and looking at the industry is building this multidisciplinary team where, well, first of all, Garrick himself has had experience building both these low-level models and high-level coarse grade models. So he's built this actin-myosin formation simulation, which I had a chance to take a look at academically, which was quite involved, which is a high-level abstraction. And we also have a team, one person, Jonathan Carr, who's actually made the first full cell model on the pathway simulation 10 years ago. He was one of the original people in Formic Labs, which is our kind of first part of a company on our side. So we have, the idea was to bring these researchers with different levels of experience, of knowledge in these different domains, and see how they can collaborate and integrate. And the ideas, as Garrick described, is that these different models and systems will support each other. And I think the other key aspects which makes this company unique is we are thinking about it as a platform. So with Garrick's leadership, we have this really truly unique research nuggets of really good technology where we can be state of the art in certain parts of discovery. But in the same time, we're thinking about it as a platform where we have general compute resources, general infrastructure where these algorithms can run, and we can provide, whether it's through Jupyter notebooks or simulations or easier tools, broad accessibility, and also integrate with these later stage or different experimental tools. So all the kind of the data sets and infrastructure is available across the board. So the idea of the company then is to provide this platform and framework where different scientists can collaborate within the same environment, and in the future even provide their own plugins and tools. So no matter how good we are at simulation, no matter how good we are at any part that we do, it's a very broad industry. So we'll want to integrate with other tools, but also provide ability for people to have components or aspects of this, which which we did not create. So as a part of a vision of a company, we want to build this more open ecosystem around making some of these things accessible. Of course, some aspects of state of the art will be unique for drug discovery and supporting, higher value clients. But we want to think about how can we open this ecosystem and cover this range to make it accessible.
So I don't know if it's probably easier if we had slides, but maybe you can walk us through any case study where you were able to show some sort of superior performance, let's say, in identifying known binders in drugs, if I'm remembering the slides that you sent me correctly. But maybe you could give people sort of an example of the power of what you guys have come together, so that, and I hate to say this, that's in an accessible way, but also that somebody in the industry would sort of go, aha, I get it, I understand why.
Yeah, I could give a quick example. So we wanted to test our virtual screening methodology, and there are multiple ways to test it. One way is purely structural. For example, you take a compound, you dock it into the pocket of a protein, and then if you know the crystal structure, you see, oh, how well did I do? Like, is it more or less identical to how the ligand was bound in the crystal structure, or is it in a completely wrong place? So that's one way how you could identify quality of docking, for example. But another way is actually screening, which is if I take, and that's one of the slides that I sent you, Harry. So for example, there's this protein called JAK2, and its biology, let's put it aside, but let's say it's a protein that's important in disease and you want to target it. And previous works already identified like about 10 compounds that were active with this molecule. And the question is, if we, in virtual screening, we are going to take millions or sometimes billions of potential molecules and find sort of the needle in the haystack. So the question is, if I take, for example, 10 known molecules and I mix in 100,000 random molecules, so then the dilution is 1 to 10,000. And your chance that you are gonna even, like, for example, if you randomly simply just shuffle these things and take top 10, your chance of finding and even a single active molecule is minuscule. It's extremely small. So we did a docking study like that, and we found that so again, we are mixing 10 active molecules with 100,000, and we docked and we scored using this AI-based scoring we developed, and then ranked. And in the top 10 ranked list, our ranked list, 3 were active. So that sort of shows a huge enrichment. I think when we calculate what is the enrichment, it's about a factor of 300. So if you had to do 100,000 experiments, this allows you to do 300 times less experiments, which is, I'm sure many labs will take that. So this is one way that it shows how computational drug discovery can tremendously accelerate research in the lab by just narrowing down things you have to synthesize and test in the lab.
So, but what are the, you've done that in one thing, but what are the challenges potentially in scaling up the technology for, say, more customers? Because I'm assuming you guys are taking on multiple partnerships, but you got to scale that technology up, and all of us know that scaling, doing it once is one thing, doing it at scale is always a different challenge. So what are the challenges you guys are facing while you guys are trying to do that?
Maybe Michael can take that can take that, because scaling is the life sciences platforms domain, I'd say, in our company.
Okay.
Well, there's different parts of this. Clearly on the first set of partnership customers, we do provide a meaningful amount of kind of hands-on help to make sure that all the results and everything is good. At the same time, we envision more open self-service aspect of it. So, for example, right now it's seen as we have two ways one could use our technology. One is through this partnership model where we help you, but then on the other hand, we have an ability to use software as a service. So the goal here is you will be able to come in and there'll be tools where you can run screens, which make easy use of docking and molecular dynamics on top. You would select what you want to do, and it would be in a more automated manner. So you don't need to have a hand-holding from us. And furthermore, we are looking at opening up some of aspects of it, like maybe some aspects of docking and other things, to a more general public through tools such as AI Assistant, or where you can actually, you just do a query and it'll do it for you. And of course, we need to think about which part of it, what is our business proposition there. But there is certain aspects of it which we would love to be available to the general public. So I think that's where scaling comes in, because you want people in smaller biotechs and universities to have aspects to part of this technology. Maybe they don't need to run such a larger screen, but they want to have some of the highest quality tools and make it very easy. So I think of scale as trying to provide enough of a technology to a broader public and number of users, learning from them. And then, of course, you need to have the technical scale of, yes, it actually needs to run lots of compute instances in a cloud to do the work. So, but that's behind the scenes on our side, and this is what we spend time on. We can do similar stuff with bioinformatics and workflows and whatnot. But
so that being the venture guy, right, it begs me to ask the question of, you've got this superpower that maybe gets you to an amazing therapeutic compound. Why wouldn't you use the technology to take one of those forward yourself and become a drug company versus make the tools available to someone else? And I'm just wondering, I'm sure you guys have wrestled with this, so I'm just curious how you guys got to where you got to, or have you gotten to where you've gotten to, or is that still being discussed?
Well, we are in the early stages, and at the same time, this is a very good question. So currently we do some lab work, and we do have plans to take certain compounds a certain distance, but we are thinking more of partnerships and a platform at the time being. And quite frankly, this is the dilemma which every biotech platform company has had to face. We have really smart scientists who really know what, like I said, are excellent at certain domain. And then we're going to build a platform, and now we're going to have therapeutics. Now, the challenge with therapeutics is it does take a fairly long amount of time. There's ID, there's all the wet lab work, and ultimately there's a clinical stage. So you're going to need to raise tens upon tens to hundreds of millions of dollars and $1 billion in the later stages, potentially in this clinical trial. And then you have investors who are coming in. Now, first of all, it's a probability game, because like I know it's like 1 in 10 of drugs gets approved. Hopefully with our AI and the tech, I think it'll be better, but it's still a chance, right? So you need to have multiple programs. But at the same time, if an investor is putting all this capital and 90% of the capital is going into a few assets, which will make or break the company, their investors and the board's focus is not going to be on the technology as much. It's kind of done. Once you have your molecule or whatever the asset is, that is your goal. right? And that creates fragmentation in the industry in the sense that there's lots of great platform companies which are started and they try to head towards therapeutics, but yet they're siloed from each other, and the technology is not shared as much, which to me, as a person who looked at the complexity of these systems and life sciences landscape in general, it feels that it limits collaboration and we're not moving as fast. So I think maybe kind of a lucky situation that I am in is I can contribute part of my capital to supporting this, which gives a little bit more flexibility and maybe in some other startups to think in a more platform and more open manner. Now, so that means we can take risks to see if certain aspects of tools will get adopted and used broader by the public, and experiment with both SaaS and partnership and some of these open business models. And in the end of the day, maybe 2 or 3 years from now, we'll decide we want to take some of these assets further down. But right now, a big part of it is trying to figure out how do we have the best possible tech and later on open it up, some parts of it, and make it more universal and collaborate more with others. That's the vision, because I believe that platform company needs to exist in this space.
Yeah, well, started my career in platforms, so I totally agree. But you guys are in a rapidly evolving space. I mean, there's 100 new papers in AI like a day, it seems like. It's some crazy number, right? So in the evolution as it's moving forward, how do you plan to stay at the forefront of understanding what's happening, developing it, and incorporating it into your platform so that it's always, the revision is staying up with where everything is going?
Yeah, at least on the simulation side, that's a challenge, and it's a very rapidly evolving field, and it's easy to fall behind. So one of the things we do is we try to stay current as much as we can with papers. Maybe not, I don't know if with all AI papers, but in particular with AI papers that are relevant to drug discovery and simulations and biological modeling. So part of our time is to actually have discussions of these papers, and we don't need to react to maybe most of them, but sometimes some papers come out that does make you question sort of your way of thinking about problems, or you realize that, oh, like here's an idea that we could maybe use in our algorithms or in our way of approaching things. So far, I feel that we've been able to keep up with that. The AI in drug discovery and biology is a smaller subset of AI, so there is more chance that we should be able to do that. But that's an excellent question. It does feel sometimes your head spinning that it's almost like every 2 weeks it's a new world, or every month. So definitely a significant focus should be on that, not to fall behind.
Yeah, I find myself like once every week or so, I'm like, no, you can't do that. Like, wow, and I have to like recalibrate myself depending on what I'm looking at. But okay, so looking a little bit into the future, what's the next big milestone for the company? How are you measuring success, let's say, or either one of those? What's the next big milestone, or how are you measuring things are moving forward in the right way?
I think on a business side, there's kind of 2 areas. One is partnerships and customer relationships on drug discovery, essentially the number and scope. And on the platform side, it is users, either awareness, if it's going to be an open tool, which is something we'll be experimenting with shortly, and direct customer use on the platform in terms of SaaS users. So right now, I would say we are still in an early MVP side for some of the life science platform tools. So those are going to be the key metrics.
So if somebody's either a partner or potential future customers, how do they keep up to date on what's going on, and how do they engage you? Is that just going to the website and clicking a button, or just curious?
Both. I think clearly having a direct email can we can email me and can email Garrick, but also we have a couple of business development people on our team. It's one way. They can read about our technology on our website. We actually just recently launched a newsletter, which is going to be monthly, and it has a bit about us, but also really interesting industry news around our domain. And so the way to do it is really go to our website and sign up for the trial for some of the tools, and or email us directly about partnerships or any questions they have.
Guys, have I left anything out? Is there any? I tried to cram everything I could through the presentation you sent me into my brain, but I might have missed something. Is there anything you guys want to touch or highlight that people should be aware of?
Yeah, maybe I could just, Michael, sorry for interrupting you for a second, but a shared vision that Michael and I have is to do wholesale simulations and bring all this technology that, I don't know what time scale, 3, 5 years, 10 years, I don't know. But to be able to take a human cell, a T cell, B cell, some other cell that are important in disease, and do basically a physicochemical 3-dimensional simulation of that cell, and then you could maybe add drugs and see how the cell responds to it. All these things that you that today you observe under a microscope, could we do it in a computer? The answer is that today we cannot for human cells, and because of that, I claim that we still don't understand biology in a fundamental way. But when that will be possible, that will be completely revolutionary. And that's one of the things that I think drives both of us, and also the parts of a company that help to basically bring that vision closer. Michael?
Yes, that
would be.
I come from the graphics space. Clearly, it's been done Oculus and the gaming before. So idea of being able to fly into the cell and keep zooming in up until a point where you see like the mitochondria and all these spinning things, and maybe pause it and speed it up and have it actually be physically accurate in real time or faster or slower seems incredible. So then you can take any measurements on top of it and it would be phenotypically accurate. That would be an incredible vision. So I think as I got into this, when you come from more gaming as a software background, it seems intuitively like it should be simpler. Why can't we just simulate it? And of course, then you realize there's quantum physics, there's just this full scope of detail is such that we haven't yet approached it, being able to have that compute capacity. But I think that's what's so exciting both about the AI and the data processing capability and instrumentation we're starting to get now is like we can get further and further down this path within a decade. And that's, you can simulate a lot of things. We can collect enough data represented. And I'm excited to basically be able to see this cell and play with it and really empower scientists with it, but also really provide tools which enable others to build this kind of things, whether it's bioinformaticians, whether it's around genomic analysis, whether it's about any data processing, to bring it together to make those models. So I think that's super exciting.
Yeah, I keep hoping that it doesn't take so long. I know that it does. Every time I think it's not going to take that long, it takes at least twice as long. But and every time we think we know something, I realize we're just scratching the tip of the iceberg about what else we don't know. So it was great having you guys on the show. I mean, obviously I'm going to keep up to date on the progress of the company. It's already done phenomenally well since inception, and I am going to make the grand assumption that it's going to continue along that trajectory and continue to do well, and change how your partners are achieving their goals of identifying new therapeutic targets and moving those forward through the value chain. So good to have you both on the show.
Yeah, it's a
Well,
pleasure.
thank you. It was really enjoyed the show, the conversation.
Thank you.
Thank you.